The first steps of filtration occurs within the renal corpuscle, which is broken down into two components, the glomerular capsule and the glomerulus. First, let's discuss the glomerular capsule. It is also known as the Bowman's capsule. Its outer wall is lined with capsular epithelium. It kind of acts like a sac that surrounds the glomerulus. Now, let's go back to the glomerulus. The glomerulus is broken down into two subcategories, mesangial cells and glomerular capillaries. Mesangial cells are basically just the connective tissue that is in between the glomerular capillaries. The glomerular capillaries is where the process of filtration actively begins. To begin, the renal arteries receive blood from the heart. Then, that blood enters the afferent arterioles. Next, the blood enters the glomerular capillaries. The glomerular capillaries are a bit more complex than your regular capillaries. Glomerular capillaries are covered by a layer of visceral epithelium. Within this epithelial layer are special cells called podocytes. These cells have feet called pedicles that intertwine with each other. The space between adjacent pedicles is called filtration slits. Glomerular capillaries are also unique in the fact that they are classified as fenestrated capillaries. This means that the capillary endothelial cells have pores that are large enough for water and solutes to pass through, but too small for red blood cells. This means, as blood is passing through the glomerular capillaries, blood plasma and solutes are going through the pores and passing through the filtration slits into the Bowman's capsule. However, there is still hematocrit in the glomerular capillaries since it is too big for the pores. The hematocrit continues to travel through the glomerular capillaries and eventually leaves through the efferent arterioles, where it will go back into circulation. Just like most body systems, glomerular filtration is driven by hydrostatic and colloid osmotic pressure in the glomerular capillaries. In glomerular filtration, there are two types of hydrostatic pressure, glomerular and capsular. Glomerular hydrostatic pressure is the pressure that pushes water and solute molecules out of the plasma and into the filtrate. Capsular hydrostatic pressure is the pressure pushing water and solutes out of the filtrate and into the plasma. The other pressure involved with glomerular filtration is colloid osmotic pressure. The colloid osmotic pressure is the pressure drawing water out of the filtrate and into the plasma. Once you have your hydrostatic and colloid osmotic pressures, you are able to find the net filtration pressure. This is the average pressure forcing water and dissolved materials out of the glomerular capillaries and into the glomerular space. Ultimately, the goal of glomerular filtration is to produce filtrate, which is a solution that is pretty much protein-free. How we typically measure how much filtrate the kidneys make is the glomerular filtration rate. This is a measure of how much filtrate is produced per minute. Typically, an adult makes about 125 milliliters per minute of filtrate.